Welcome to my channel, Sunlight Shine. Today we are going to learn about DNA deoxyribonucleic acid. Basics. Deoxyribonucleic acid, more commonly known as DNA, is a complex molecule that contains all of the information necessary to build and maintain an organism. All living things have DNA within their cells. In fact, nearly every cell in a multicellular organism possesses the full set of DNA required for that organism. What is DNA? DNA, or deoxyribonucleic acid, is the hereditary material in humans. DNA is a polymer composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth and reproduction of all known organisms and many viruses. The discovery of the DNA, the twisted ladder structure of deoxyribonucleic acid, DNA, was discovered by James Watson and Francis Crick, marked a milestone in the history of science and gave rise to modern molecular biology, which is largely concerned with understanding how genes control the chemical processes within cells. The four scientists that discovered DNA. These four scientists, Crick, Franklin, Watson, and Wilkins, co-discovered the double helix structure of DNA, which formed the basis for modern biotechnology. Scientist Maurice Wilkins and scientist Rosalind Franklin. Scientist James Watson and scientist Francis Crick. The two DNA strands are known as polynucleotides as they are composed of simpler monomeric units called nucleotides. 2-3, each nucleotide is composed of one of four nitrogen-containing nucleobases, cytosine, C, guanine, G, adenine, A, or thymine, T, a sugar called deoxyribose, and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds, known as the phosphodiester linkage, between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar-phosphate backbone. The nitrogenous bases of the two separate polynucleotide strands are bound together, according to base-pairing rules, A with T and C with G, with hydrogen bonds to make double-stranded DNA. The complementary nitrogenous bases are divided into two groups, pyrimidines and purines. In DNA, the pyrimidines are thymine and cytosine, the purines are adenine and guanine. The above figure is their chemical structure of DNA, hydrogen bonds shown as dotted lines. Each end of the double helix has an exposed 5-phosphate on one strand and an exposed 3-hydroxyl group, O, on the other. Congratulations! You have gained enough knowledge today.